Before starting this lab, I first created my disinfectant solution using 10% bleach and water, spread it across the surface, and made sure it sit for 2 minutes before cleaning it up. Currently in the video, I made sure the particles of my broth solution was broken up, and now I'm currently getting the sterile loop out of the package to proceed with the lab. I'm now picking up my triptych soy broth and being sure not to sit my loop or cap down. I will be heating the top of the tube to make sure it is not contaminated. I rotate it 3 or 4 times under the heat. Then proceed to tilt my tube and put the loop in to gather some of the broth onto the loop. Before capping the tube, I put it under the heat to make sure it stays uncontaminated. While still holding the loop, I pick up my nutrients broth. Holding the cap in my hand, I heat the top of the tube to prevent contamination. I again tilt my tube to put the loop in. This is done to transfer the triptych soy broth containing the bacteria into the nutrients broth. Again, the tube is heated before placing the cap back on to prevent contamination. Once placing the tube back, I place my loop into my plastic cup for sterilization. Moving on to activity 3, I'm getting out my sterilized loop. I will now uncap the tube of the triptych soy broth and heat the top to prevent contamination. I will tilt the tube at an angle and place the loop inside the tube to gather some of the triptych soy broth. The tube rim is heated again and the cap is placed on. While still holding the loop, I pick up the triptych agar slant. Uncapping the tube and heating the tube rim to prevent contamination. I proceed to slant the tube to spread the triptych soy broth onto the agar slant. I slowly start at the bottom and work my way to the top to spread the broth on the slant. Tube is heated and cap is placed back on. The loop is placed in the plastic cup for disposal. I take out my sterilized loop for activity 4. Moving on to activity 4, I pick up my tryptic soy broth, heat the tube, tilting the tryptic soy broth, I place the loop inside to gather some of the broth. I again sterilize the top of the tube and place the cap on. I uncap the Petri dish, keeping the lid at an angle to prevent air contamination, and slowly work the loop down the Petri dish in a zigzag line. I then proceed to place the lid back on and flip the dish over. This is the end of my video for this lab. Sorry, I forgot to record the lab area disinfection. But after disposing of all materials, I repeated the same process for disinfecting the area surface and then wiped it off with soap and water afterwards.